Hello everyone, and I am back with another video, that is ELISA. So I hope you guys are doing well, and it's a immunological technique called ELISA. So the full form is enzyme-linked immunosorbent assay. As the name suggests, it is something linked with enzyme that further shows you colored substance by which you can detect or analyze quantitatively or qualitatively. Okay, so in ELISA we use antibody and link it with enzyme, and you can also say it is an enzyme conjugated antibody. So as it named, like enzyme linked immuno here means antibody. So antibody is used with enzyme, or it is linked with enzyme, so that it react with colorless substrate or chromogenic substrate, and this enzyme or substrate gives you colored product. For example, the enzyme used in ELISA are alkaline phosphate, horseradish peroxidase, beta galactosidase. So with a specific antibody, when a specific antigen binds, and with that antigen we will link an enzyme, for example alkaline phosphate. So this alkaline phosphatase or PNPP, that is para nitrophenyl phosphate, when come in contact with enzyme, any one of the three, though, so it gives a colorless and product, so yellow color product formation. This will allow you to uh, to measure the concentration. There are three type of ELISA. First, I'm going to tell you about indirect ELISA. What happens in indirect ELISA is that given sample, you can detect antibody. Okay, so basically it you will use antigen coated well, and you will wash it, and then you will add specific antibody. So you will come to know that there will be antigen antibody interaction, and again you will wash it out, and then there will be a secondary body that bound to the primary body, and the enzyme conjugated with the secondary antibody. Okay, now addition of chromogenic substance, for example PNPP, this para nitrophenyl phosphate, it is a colorless substrate or chromogenic substrate. So when it is linked with enzyme, it gives a product which shows color. So you can measure the amount of color. So amount of colored reaction product measured by spectrophotometric plate reader. Okay, spectrophotometry work on the principle of absorbance. So higher the absorbance, the greater is the concentration. Okay, uh, and what is the purpose of giving or adding secondary antibody is that you can check whether the primary antibody binds to it or not. Okay, now in sandwich ELISA, as the name suggests, something is in between two of the component. So in the indirect ELISA I am using antigen coated well, however in the sandwich ELISA I am using antibody coated well. So I am going to add specific antigens to it, so my antibody will be primary antibody, and you will come to observe antigen antibody interaction. So I am going to wash the well now. So again I am going to add my enzyme, that is alkaline phosphate, for instance. So this will make a sandwich like appearance as primary antibody here, and then secondary antibody which is merged in between, yes, in between antigen. Okay, and then enzyme on it. Again addition of chromogenic substance give what colorless colorful product. So uh, as a result, the absorbance of the color can give you the concentration of the antibody. So indirect is used for antigen, and sandwich is used for sandwich in which you coat the well with antibody. Competitive ELISA is that you will use antigen coated well, and for it is a determination of antigen concentration. So in one case you use an unknown sample that has no antigen. So since it has no antigen present, your sample has, so your antibodies bind to it, that is conjugated with the enzyme, and then you add secondary antibody. Okay, and wash it with and then add PNPP, then you will observe the color. And you this when your sample has no antigen, so high absorbance is observed. In second case, when your sample has medium concentration of antigen, so you will use a antigen coated well and incubate antibody with antigen to be measured. So here you see that antigen antibody mixture is obtained. However, some of the antibodies do not bind to it and antigen remains free. So now the enzyme link conjugated antibody is added, and then addition of P and PP. This will show you less color. So low absorbance is shown because there are no free antibodies or antigens left in the well because of previously having medium concentration of antigen. Okay. So high absorbance means no antigen and low absorbance means antigen is present. Okay. So this is what ELISA and its type. Now I am going to tell you about ELISA spot. ELISA spot assay. It is basically measured for, or it is a modification of ELISA assay, or you can detect cytokinin secretory cells present in the suspension or not. So basically it is for a particular type of measurement of cytokine secretory cell. So you will add non-secretory or secretory, both the antigen into the well, so well coated with anti-cytokinin antibody. So here the antibody is anti-cytokinin, so secretory cells bound to it and relieves the cytokine. However, non-secretory cells remain unbounded, and when I wash it, so again I linked with my secondary antibody with the enzyme, and this antibody is a cytokine. Okay, then you will measure the color, and this is what secretory cell population look like. Okay, and however the blank space is non-secretory cell population. Radioimmunoassay is RIA. It is something when used for the measurement of drug, hormone concentration, vitamin concentration, or protein concentration in blood. Okay, most sensitive technique since we are using the radio labeling. Okay, so radio labeling is done by 125 iodine, that is gamma emitting isotope, and hydrogen tritium, that is beta rays emitting isotype. So in case 1, unknown sample with no concentration of antigen. So when your antibody coated well is there, so radio label antigen come and bind to it, and all antigen bind to radio label all antibody bind to radio label antigen. So radioactivity is 100% when no antigen is present. However, in case 2, high concentration of antigen is present, radiolabeled antigen, and unknown sample having unlabeled antigen. So you will see that antibody come and bound with both unlabeled and radiolabeled antigen. So this is the graph that with the increasing concentration of 125 I antigen bound to antibody, there is a decrease in concentration of unlabeled antigen. Okay, so with the decrease in radioactivity, there is an increase in unlabeled antigen. Okay, so this is radio immunoassay, and I hope you like my video and understand the concept of ELISA spot and ELISA. Thank you.